This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hello everyone, and welcome back to Beyond Cancer. As part of our 2024 Cancer Neuroscience Symposium series, I'm here with my co-hosts, Dr. Brandi Mattson and Dr. Amit Moran to interview Dr. Brian Davis.
Hey, thank you, Sahana, and thank you so much, Brian, for joining us today. Dr. Brian Davis is a professor of neurobiology at the University of Pittsburgh. Dr. Davis is one of the giants of the field. A lot of the papers that established the field of cancer neuroscience stemmed from his lab. Many of the speakers and many of the key opinion leaders in the field graduated from Brian's lab. Brian is heavily invested in cellular and molecular mechanism for central and peripheral nervous system development and plasticity, but in our world, or in the context of our work, Brian's work was mostly focused on peripheral nervous system plasticity in cancer, which is a fascinating, new, and rapidly growing field. So it's great to have you with us, Brian. We're really looking forward to hear about your work. And we're looking forward also to see you in Houston in the meeting.
Happy to be here.
Can you tell us a little bit about, yourself, your current lab research?
Sure. So I've spent most of my career studying the biology of the peripheral nervous system, including the sensory nervous system, the autonomic nervous system, sympathetic and parasympathetic, my enteric nervous system. And as a faculty member, my first really well-developed research program was looking at growth factors and their role in the development of the peripheral nervous system. This is work I did in collaboration with my wife, Dr. Catherine Alvarez, who was a skin biologist, and she had done for her postdoc, she had been looking at the keratin genes in the skin and their role in development and regulation of skin homeostasis. And the cool thing was that she had known, she was in a laboratory of Elaine Fuchs, and Elaine Fuchs's lab was one of the first ones that identified the structure of keratins and the promoters that drive the different keratin expressions in the different epithelial tissues. And Kathy knew the promoters that could direct the expression of growth factors to the skin, neurotrophic factors to the skin. And it was actually Kathy's idea to put the NGF family members, the GDNF family members, under control of these keratin genes so we could overexpress them. The skin normally expresses these growth factors, but Kathy's idea was to overexpress them. Luckily for me, she wasn't a neuroscientist. So she needed a neuroscientist who could actually interpret what the heck happened when she overexpressed these growth factors in the skin. And that essentially led to an odyssey of around 20 years in which we made the different transgenic animals overexpressing these growth factors to see what effect it had on sensory neurons innervating the skin, sympathetic neurons innervating the skin, and it even led to later studies where we expressed growth factors in other epithelial structures like the colon, where we actually looked at what that did to innervation of the gut. And it also dovetailed with the arrival of optogenetics, which allowed us to target expression of various things like chenorodopsin and GCAM to the different structures in the peripheral nervous system. So that's sort of where I started in terms of getting into the cancer area. Once we had done a lot of work on looking at the development of the peripheral nervous system, we realized that these same tools could be used to study diseases of the peripheral nervous system. And a lot of these growth factors, in particular the NGF family members, were involved in things like pancreatitis or inflammatory pain conditions. And so we started studying the role of the growth factors on the sensory side and their role in inducing pain in models of like pancreatitis, in models of inflammatory pain like with CFA, complete void adjuvant, typical sort of pain, peripheral pain models. And that was very productive. We learned how inflammation changes gene expression in sensory neurons and leads to chronic pain. But then in the 2000s, we moved from the University of Kentucky to the University of Pittsburgh into the GI division, where we got a crash course in diseases of the gut. And we realized that a lot of diseases of the gut are inflammation-based. And we also knew about the potential role for sensory neurons in driving these inflammatory conditions like chronic pancreatitis. And we did a number of studies showing that activation of sensory neurons in models of pancreatitis could drive a neurogenic inflammation driven by the immune system, and that by modulating the sensory neurons, we could actually block the disease. And it was about that time that Andy Rim came out with his paper showing that inflammation is a major contributor to pancreatic cancer. And Andy had a nice little piece of data showing that if you block the inflammation associated with his genetic model of pancreatic cancer, he could actually slow or halt the progression of the disease. We knew that sensory neurons could play a major role in inflammation in the pancreas. And so Andy came here and gave a talk, Andy, who's now at MD Anderson. And I met with Andy and said, hey, let's denervate the pancreas. And we thought it would have some mild effect on the progression of cancer. And this is work that I did with, again, with Kathy Albers, Rochelle Stopinski, and Jamie Solomon. And what we found to our surprise is that in a lot of the animals, the cancer just never developed. And this is in a genetic model of pancreatic ductal adenocarcinoma, in which virtually all the animals get cancer within 6 to 8 months. When we denervated them, removed the sensory input to the pancreas, the animals were living for a couple of years with no evidence of cancer. And this sort of blew our mind. Again, initially, we put this down to being sensory neurons playing some role in driving the inflammation that was driving the cancer. Now we're not so sure. But that's sort of how I got into this area of the role of the nervous system in cancer. Right. And that's, I think, one of the more important questions and issues that the field has to deal with, right? Because so many of us who look at this tend to underplay the complexity of the peripheral nervous system, right? And so every one of these organs that you've mentioned, any one of the peripheral organs, any structure that's been looked at, they're innervated by sensory fibers, sympathetic fibers. They're going to be innervated by some component of the parasympathetic nervous system. All these things are there in different mixtures. And like you say, each tumor is different. And even within an organ, each type of tumor is going to be different. There is no one type of pancreatic ductal adenocarcinoma. You can get there a bunch of different ways, and the transcriptomic, the proteomic landscape is going to be different for each tumor, for each individual, and you may even have multiple types of tumors in a single organ, in a single patient, and the nerves are going to be responding to these differently. It's really fascinating because we tend to think of nerves, and especially like the sensory neurons, we tend to think of them as that being their job, that they're detecting the environment, that they're sensing what's going on in the environment. I think that might just be a side gig for a lot of the sensory neurons. In fact, sensory neurons release all kinds of molecules in the periphery. They are talking. There's a conversation going back and forth between them and the developing tumors. The sympathetic neurons that are running with the sensory neurons are also having a conversation back and forth with the tumors. And to make things more complicated, they're talking to the sensory neurons. And one of the other things that blows my mind is that when you look at the phenotype or the gene expression pattern within the tumors, for some reason that I don't think we appreciate, actually I know we don't appreciate, tumors adopt this neuronal phenotype. I mean, what is up with that? What do they think they're doing? I mean, tumors express neuronal growth factors. They express the receptors for those growth factors. I mean, why are tumors expressing TrkA? TrkA, one of the most important tyrosine kinase receptors for neural development, is expressed by tumors. And in fact, TrkA was originally identified as a gene in a prostate cancer. We're really not appreciating that these cells have a lot more in common than they have differences. And I think trying to understand what you're talking about in terms of the landscape is really the challenge. It's the next challenge. And if you look at the way the field is developed from the initial studies from Paul Fournette, who was actually really focused on autonomic innervation of his genetic model of prostate cancer, where he identified a particular role for both the sympathetic and the parasympathetic nervous system. And then you have Dr. Wong come along and look at gastric cancer, where he's looking at the role of the parasympathetic system and cholinergic signaling driving that cancer. And then Jamie and I are saying, no, it's the sensory neurons. I think in every cancer, people have been able to find a rationale why different parts of the nervous system are involved. And to be fair, each of us ignore the other neurons, the other kind of axons or nerve type that is in our nerve that we're either cutting or chemically lesioning. And we all sort of pretend like we found the one cell type that's important when they're probably all important. And nothing can happen without all of them interacting in some very complex and dynamic way. So, that's a long way of saying, it's a long way around to saying that I have no idea how to answer your question because I think we're really just at the beginning of this.
Yeah, and I think we found some sort of a creative solution for that. We're just saying that those nerves are switching roles, as you probably read in our head-and-neck tumor, publication.
Yes.
But yeah, it's definitely complicated. But, going off of that, pushing that question a little more, how do you think we can harness the nervous system to remodel the neural tumor microenvironment?
Yeah, so, no. I don't think we know enough at this stage to be thinking about harnessing the nervous system to actually change the tumor microenvironment. Because it's really a chicken and egg question, right? We don't know who's going first. We wrote it in our review chapter in the book that Moran edited with Nicole Sheff. We're proposing that maybe one of the major roles for the nervous system is essentially setting up an environment that's permissive for the development of the tumor. In other words, that, we have this idea of what we're calling the safe harbor, where because the peripheral nervous system does not divide in the adult, the peripheral nervous system that you have once you're born is a peripheral nervous system you're going to have for the rest of your life. It's got to stay out there and survive all the horrible things we do to it as an organism. All of the inflammation, all the alcohol we drink, all the toxic compounds we take in, all those things that can activate the immune system, and these poor nerves have to stay out there in the tissue trying to keep everything going forward. So they're under a constant assault from all of the, environmental things that are happening, plus an immune system which gets pissed off from time to time and starts attacking everything. So, one of our thoughts is that neurons require some way to dampen down these immune responses. And by doing so, might actually block immunosurveillance. And that might set up an environment that would be conducive for the development of tumors. So that's just one idea. And that the nerves might be actually at the front end of cancer, not so much necessarily driving cancer once it goes. But there's a lot of data out there, obviously, that neurons could also contribute to the evolution of tumor genesis. So obviously that's where you're thinking, okay, so how can we manipulate the nervous system to halt the cancer? All of the studies that we've all done where denervation does something and seems to slow the growth of cancers, those are most efficacious. Those kind of manipulations are efficacious at the beginning of disease. We really haven't explored to a great extent what we could do once the disease is established in terms of manipulating the nervous system to change the trajectory, the course of the disease. And I think that's going to be more difficult. If you want to go there, now we're getting very, speculative, sort of Star Trek-y, because nerves are pretty wonderful things. As we sort of pointed out in our review chapter, they're everywhere. There are very few cells in your body that are more than a couple of cell diameters away from some kind of neuronal process. We don't really appreciate that because histologically, when you look with the tools that we normally use, we often underappreciate how densely innervated our tissues are. A lot of the tiny little fibers of the peripheral nervous system are really right at the edge of what you can see with light microscopy. So most of us don't appreciate how intensely we're innervated. We are very intensely innervated. So that does provide the opportunity to maybe manipulate those fibers to have them have some kind of impact on cancers, on developing tumor cells. So the obvious things are obviously increasing or decreasing their activity, their excitability. So you can do that, and we already have good data that shows that even things like potentially turning down the sympathetic nervous system can be therapeutically productive. All right, so that can be a good thing, but that's pretty gross. I think what you're trying to think about, and what many of us are sort of fantasizing about, is our ability to get novel genes or small molecules expressed in nerves and have the nerves target that to the tumors. I mean, that would obviously be super cool. I think there's a lot of people out there that are developing viral mechanisms for delivering constructs that will get expressed in sensory neurons. I don't think people are thinking about that in the terms of cancer per se yet, but there's a number of therapies on the horizon for treating other genetically based diseases that are primarily that are neural targeted. Some of the best viral vectors are neurotropic. In other words, they love to infect neurons. That gives us an opportunity to get things expressed. But we are so far away from doing that. But you can imagine at some point, you go into the doctor's office. They would know which part of your nervous system they want to target. We're going to express a gene there that'll then be delivered to the site of where the tumor is growing. Or in the case of metastasis that spreads in the nervous system, something like that could be quite useful in terms of trying to slow a cancer. We're way. I mean, there's one thing to talk about personalized medicine, and there's another way to talk about cell-specific personalized medicine. So yeah, we're not there yet, but maybe one day.
Yeah, I completely agree, and we are way behind cancer in our ability to manipulate nerves in a way that would be beneficial for the host, for us as patients. I was wondering if in your lab you're looking at the factors, the cancer-derived factors that manipulate the nervous system. Because it seems like you mentioned Dr. Fermet's work and Shaw's work on how specific branches of the nervous system contribute to cancer. But in many instances, we see that in a similar process to angiogenesis, those nerves are growing in tumors, and sometimes, as we described a few years back, might even acquire new features. So, how do cancer doing that? Do you have any cool data to share with us on it?
So this goes back to that whole question. I mean, this started way back when from the German group that includes Ekin Diemer, who's going to be at your meeting and hopefully at the Keystone meeting. Well, definitely he'll be at the Keystone meeting. We've known for a long time that tumors like to make growth factors that directly affect the peripheral nervous system, and it makes them sprout, and it brings them in, and they grow into the tumors, and sometimes quite dramatically. And of course, this provides a substrate for the tumors to then migrate into the nerves and spread throughout the entire body. Why the heck tumors do this is still a mystery, but it seems to be a common feature of many, many different types of tumors. And it goes back to this idea that for reasons that aren't clear, a lot of tumors begin to assume a neuronal phenotype. They start expressing all kinds of voltage-gated sodium channels. They express calcium channels that you would more normally associate with neurons. And they do weird things that make it look like these are neuronal wannabes. Neurons don't normally make their own growth factors. They make the receptors. And as I said earlier, the tumors are making both. But it's very clear that there's this cozy relationship that develops between the tumors and the nerves in such a way that they seem to become co-conspirators in the development of the pathology. Why they do this, again, I don't know. We never want to assign a conscious property to a tumor. It's really more of a statistical phenomenon that certain cells are going to divide out of control, and other cells that divide out of control, some are going to do better in terms of going towards actually becoming a tumor than others. And maybe part of that program that statistically allows them to become a successful cancer. In other words, one that doesn't get squashed by the immune system, or one that just sort of doesn't peter out because it can't establish, vasculature or whatever, are those tumors that develop an intimate relationship with the nervous system. Because the nervous system can provide a lot of good things to a growing chunk of tissue. One of the things that I go back to is one of the earliest findings about the importance of nerves and development. Back in the day when I was an undergraduate in my honors biology program at the University of Illinois at Chicago, there was this phenomenon that had been known going back way when, and actually David Stockham was one of the guys who studied as a developmental biologist, that in a salamander, if you cut off their leg, they will regenerate it. They develop a blastema, which is this undifferentiated mass of tissue that's at the end of the limb, and then you'll grow a whole new limb. It's really cool. But if you denervate that limb, they never regenerate. So what the heck is it about the nerve? What is the nerve? It's a nerve. What is it bringing to this massive dedifferentiated tissue, this blastema, that allows it to grow a whole limb? There is something intrinsically coming out of the nerves. And for a long time, it was thought to be the electrical activity, that somehow the electrical activity, and this led to a lot of really bad science, looking at magnetic field and electrical activity, trying to induce regeneration of the spinal cord. Overly simplistic, didn't really go anywhere. But we still have these really foundational observations that the nerves are really important when you want to grow new tissue in the adult. And I think that there's something telling there in that experiment that would be informative, or is telling us something about why nerves and tumors are so intimately related. We just haven't unraveled that yet. That's where, and that's going to involve growth factors. It's going to involve all of the adhesion molecules that are normally there during development. All those pathway finding molecules. There's stuff going on there that we are just scratching the surface right now.
Yeah, I cannot agree more. We have very little understanding on the roles of nerves, in general in health and in cancer. But we definitely know that they're growing into those tumors. And when you're looking at clinical trials, you see that there are nearly 4,000 clinical trials looking at angiogenesis, 0 looking at neurogenesis, even though we all know it's a foundational, critical feature of human development and tumor development downstream to that.
Yes, of course. So we wanted to just ask, because we were actually really interested in this, but how has optogenetics kind of transformed our ability to study neural circuits? And, what challenges have you encountered when you were applying this technique to studying sensory neurons?
What to do first? So optogenetics has changed everything. It's tough to be a neuroscientist today if you don't do optogenetics. So optogenetics is a really big term, right? So when you say optogenetics, I think most people think about channel rhodopsin, halo rhodopsins, the novel opsins that allow you to increase activity or decrease activity in a population of neurons, usually combined with a transgenic expression paradigm. So you express it in a particular type of cell that's activated by light of different colors. That's only half of it, or a third of it, because then there's the G-camp and the part of optogenetics where expressing light-activated genes that allow you to monitor activity, and the voltage-sensitive dyes. So optogenetics, you can turn things on, and then you have the whole G-camp and expression of molecules that allow you to monitor activity. And then there is the other part, a third part of optogenetics, where you can use light to induce protein aggregation or bring genes together to actually initiate an enzymatic process. Optogenetics is huge. And of course, when you think about optogenetics, there's also the other genetic strategies, like the chemogenetic strategies, where instead of using light, you're using novel molecules to turn on genes in a particular population of cells. It's a candy store. It's a gumball machine of options, and what do you do first? And so, for us, it has allowed us to ask specific questions. For example, what happens when we stimulate sensory neurons? How does that activate a particular tissue? What does that do to, markers of inflammation? It allows you to do things where instead of trying to induce a disease to activate neurons, which has a whole host of changes that are associated, if you were to, in a model of pancreatitis, try to induce pain, you're doing so many things, it's hard to dissect what's doing what. But with optogenetics, you can actually just turn on the sensory neurons going to the pancreas and ask specifically what are the sensory neuron activity does in terms of the disease process in the pancreas. So it really allows you to dissect things at a much greater resolution than you could previously when you had to induce the entire menu of things that happen, all the changes associated in a disease model. It's revolutionary. I can't say that enough. And it's just changed everything, and the way that the field has gone. It's been incredibly collaborative. People have shared the animals that express the different genes under different promoters. And, when you combine those now with the genetic models, the sky's the limit. The genetic models of cancer. So you can, for example, express some of these opsins, these novel opsins, in, the model of pancreatic cancer, the genetic model of pancreatic cancer. And then you'll be able to turn on genes, we'll be able to turn on sensory neurons or sympathetic neurons while the animals are developing tumors and actually see how that drives or inhibits progression of the disease. We're going to get some very clear answers putting all that stuff together, especially now if you mix in CRISPR technology that actually allows you to get the multiple novel genes expressed all in one animal. I think that's what's slowing us down up till now. If you have to read all these genes in, it's a nightmare, right? If you have to read in your channel rhodopsin, you have to read in your gene mutations for the cancer, that can take you many, many generations. But by a combination of either viral expression or CRISPR, you can put all these genes in one animal and start to ask very specific questions. So, it's been a boon. It's been a boon, but it's also been almost too much, almost too much of a good thing because, you're bouncing around. You're bouncing around from one great idea to the next great idea. It's sort of hard to focus on one thing and really make progress when there's so much good stuff that you want to try to do.
It sounds like you just are a huge proponent for the excitement within this field and the future that lies ahead. To kind of focus it back down for the symposium, what are you most excited about for the 2024 Cancer Neuroscience Symposium?
So, seeing people in person, I sort of stopped going to meetings, and so many reasons not to. I mean, Zoom is great, but I've had enough. So I'm really curious to get into a room with my colleagues and actually talk about stuff that you really can't do in the same kind of way on Zoom. I really want to see where the field is. I have to say that as excited as I am, and I'm very enthusiastic about this field, I think we're at a potentially dangerous point because there's been so much enthusiasm, so much interest in the field. I think it's very important that we don't overpromise the potential of this field. We have to double down on being rigorous in our analysis and our experiments because, in the abstract that I wrote for the Keystone meeting, and they're making me take this out because they thought it might be a little inflammatory. This field has brought together oncologists, cancer biologists, who know just enough about neuroscience to be dangerous. And the neuroscientists have complementary weaknesses when it comes to cancer. So you have these people who know their field very well and barely know the other field. And we're all jumping in with both feet. And marvelous stuff can come out of this, but also not so marvelous stuff can come out of this. And I think we really need to develop a conversation to make sure that we all understand, how important it is to really appreciate the complexity in each of our fields and to come together in such a way that when we make a statement like nerves can stop cancer, that we know what the heck we're talking about as neuroscientists. And, when the cancer biologists tell us that, nerves are doing such and such, we have to make sure that they really understand what they're saying about what nerves can do and what nerves can't do, at least as far as we understand it as neuroscientists. So I really want to have these conversations, and I have a feeling once we get together and talk as a group, we're going to identify whole new areas where we should really be focusing. There may be some areas where we need to go back and really ask, was that conclusion right? Or were we over-interpreting our data? And so I'm really excited. I want to have this conversation. I want it to be robust. I want it to be a free exchange of ideas. And, I'm just really looking forward to learning a lot, because every time I talk to my friends who do cancer or immunology, oh my God, immunology, my head explodes. I mean, I learn so much, and there's so much I realize I don't understand about the immune system. And I think that's going to be a major player in everything we're talking about. I mean, that is huge. And I think, going back to the question earlier, where can manipulating nerves affect the tumor microenvironment? It very well may be that it'll be through modulating the immune system, or modulating the vascular system, or modulating, the connective tissue, the fibroblasts. I mean, those guys are really involved, and they can talk to neurons as well. It's those kind of ideas that we really need to develop more fully. And so it's very easy, as a neuroscientist, to see something that immune cells are doing and get very focused on that and ignore the fibroblast, ignore the vasculature, and get blinkered. And we sort of have to stop doing that. I think, if this field's going to go forward in the most productive way, now that we've expanded our idea that nerves are important in cancer, we can't just then become insular and just look at nerves in cancer. We need to keep the broader, the larger ideas of that it's the whole community of cells that's there that are interacting. Because otherwise, we're going to end up going down the same sort of narrow tubes that we've gone down in the past and don't want to do. We have to keep ourselves open to all the other things that might be going on. And that's where coming together at a meeting like this would allow us to do that. So that's what I'm hoping for, that kind of, productive interactions, and at the same time, that input from each other that will allow us to really see what's going to be required to make observations and conclusion, testable hypotheses that will drive the field forward, as opposed to sort of getting down in our little narrow pathway saying that our nerve cell, our particular cell, is important for this process in cancer, to the exclusion of all the other things that are going around there. I think we don't want to do that. We don't want to become segregationists as far as what tissues and what ideas are important. We need to keep our ideas or our minds as open to the possible interactions if we're going to get the most out of it.
Yeah, as a person studying tumor microenvironment for over a decade now, I see many papers that take a very reductionist angle on things. Just, I'm going to work with this cell in cancer or that cell in cancer. And I think one of the beautiful things that I've experienced in the previous meeting and the current meeting is to be able to put all those people working on those different cells in the same room and discuss. And instead of talking about cell-cell interactions, start looking at neighborhoods and communities of cells. So with that, I'd like also to congratulate you. Hopefully those meetings will merge with your meeting, with the Keystone meeting, which we're looking at starting 2025, but it's still in the works, I know. But congratulations, this is a huge achievement for the field, and thanks for your leadership with that regard. And with that, I'd like to transfer the podium to Sahana.
Yeah, thank you so much, Dr. Davis, for being on our podcast today and sharing us your insight on everything. And we are so excited, and the listeners are so excited to hear all about you. So with that, we'll finish this session and see you guys next time.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.